Wild Health is about optimizing you. We use genomics, blood work, biometrics, microbiome assessment, many other tests, and a conversation with you to come up with a full health optimization plan. What's the perfect diet, exercise, and supplement plan for you and only you? The Wild Health podcast is about optimizing all of us. Here we cover the cutting-edge science that gives you the base to be able to apply the personalized plan we give you as a patient. To sign up as a patient, go to wildhealth.com. Or if you're a physician or health coach and you want to learn how to do this for your patients, we're happy to help as well. Wildhealth.com for all the information on becoming a patient or working with us.
The absolute pleasure today of interviewing Dr. Michael Snyder. He's a professor of genetics at Stanford University School of Medicine, the director of the Stanford Center of Genomics and Personalized Medicine. As a pioneer of precision medicine, he's invented many technologies enabling 21st century healthcare, including systems biology, RNA sequencing, and protein chip technology. He's initiated big data approach to healthcare through his work using multiomics to detect early stage disease, including wearables and microsampling to measure hundreds of molecules from a single drop of blood. He's the first researcher to gather petabytes of data on individuals, which is millions to trillions times more data than the average clinician collects. He's published over 900 papers, is one of the most frequently cited scientists in the metabolic health spectrum. And in terms of commercial success, Dr. Snyder has co-founded 17 companies, which combined enterprise value of over $6 billion. Very impressive. And with that, Dr. Snyder, welcome to the Wild Health Podcast.
Oh, it's a pleasure to be here.
Yeah, so my hope is our focus today is to talk about a couple of papers that have come out of your lab focusing on subphenotypes of type 2 diabetes, which is a revolutionary concept from my perspective. But before we get into those details, I'd love to just hear some background information about how you came into this personalized medicine, precision medicine space, because that certainly didn't exist probably when you did your initial PhD training and your postdoc. So how did this come about from your perspective?
Sure. Yeah. Well, people used to study genes one at a time when I was getting my PhD. Recombinant DNA was brand new, so I'm really dating myself here. And then I think our claim to fame initially was that we started working on yeast at the time, and we looked at basically all the yeast genes, 6,000 all at once, set up methods for studying them, and then studying biological problems at the systems level. And then, to be honest, it made a lot of sense. We always wanted to apply it to medicine. And I moved to Stanford about 16 years ago and was embedded in a medical school. It really gave that great opportunity to take that same concept. Let's not just study little bits of things like they do when you go to a physician's office. Let's really try and get a comprehensive picture of people's health. And that's really what started it all. And what got me into diabetes, if you want me to go further, was the fact that we sequenced my genome, discovered I was very high risk for diabetes, which I was not aware of that. It wasn't in my family history necessarily, although in hindsight, it probably was. But my genome predicted I was at risk for diabetes. And through this high profiling, I discovered I got diabetic after viral infection of all things. And so when we picked it up right away, so that's how I got interested in the diabetes side of all this.
Wow, that's awesome. That's very interesting. I don't know that anybody would ever look at you and sort of think about diabetes as being a potential medical issue for sure.
No, I'm on the thin side. In fact, when I first showed some of my genome and why I wanted to measure a little bit more for glucose, they call it insulin resistance and things like that, the doctor told me, why would you do that? You don't have a family history. You don't look it, because I am pretty thin. And I said, well, my genome, looks like I'm at high risk and we should assess these things. And at that time, I was not diabetic, but I became diabetic shortly thereafter, actually, after this viral infection. So we actually think it's combinations of genes and environment that triggered it for me.
Yeah, that's fascinating. That's a good segue, I think, into the focus of our conversation today in terms of, diabetes. I think historically, and even, not even historically, to the present, as I talk to colleagues of mine outside of kind of personalized medicine, when you ask them about type 2 diabetes, they speak of it as a singular disease. It's a singular pathophysiologic problem. It's, got a set of treatments, and it's very clear from this paper that is not at all the case. And it's never been the case.
Yeah, absolutely true. It's like saying all animals are the same: cats, dogs, people, cockroaches. But when you really look at it, maybe to a Martian that looks true, but
Right.
to us it doesn't look true at all. So, and that is the case for type 2 diabetes. As we start digging in, it's pretty clear that you can have what's called muscle insulin resistance, meaning your muscle cells don't respond to insulin and don't take up glucose. There's liver insulin resistance, hepatic or sorry, adipose insulin resistance, so fat insulin resistance. But on top of that, there's something called a beta cell defect. Turns out that's what I have. I make insulin, but I don't release it from the pancreas. And then there's something called incretin. These are hot items these days. You may know they're the GLPs that everybody's taking, like Ozempic, some mimic of that. And so there are people who have issues there. And so what we did was we went in and started subtyping people for what their kind of diabetes is, and discovered that, some people are incretin resist incretin defects, some people are beta cell defects, as I say, that's what I have. Most people are musclins resistant, but actually the majority are combinations of these things. So you can be both musclins resistant and have a beta cell defect. And and that well, we'll get into that, but we think that turns out to be pretty important.
Yeah, that's a fascinating concept. And I think certainly for me was extremely illuminating just thinking about the fact that you have all these different pathophysiologic mechanisms that maybe give us the overall phenotype of a type 2 diabetic, but clearly have very different inputs that lead to that and therefore potentially different responses to lifestyle interventions and pharmaceuticals and that sort of thing. So let's get into the study because I think
Okay.
it's worth understanding sort of from a methodology standpoint, how the study was created, and then certainly what the findings were on the back end of that.
So what we wanted to do was see, again, maybe to back up. So type 2 diabetes, they classify it by high glucose, and period. Everybody's lumped together. And so what we did was we went and we knew there were mechanistic differences. And so we started subtyping people along the ways I described: muscle resistant, beta cell defects, incretin defects, and hepatic. And we've looked at other things too. And it's not to say there aren't other things involved, like your microbiome probably is important too. We did measure that, but we don't have that totally weighed in yet. But we went in and we first subtyped people. And then we also noticed that their glucose curves, may know about these continuous glucose monitors. You can now get them over the counter. They're very powerful. I recommend all your listeners try this thing. You'll never eat the same. And I mean that in a good way, meaning you'll see what foods will spike your glucose and which ones don't. And then it's very simple. You avoid the ones that do, and eat the ones that don't spike you. And in some cases, a lot of cases, obvious in hindsight, might be as simple as just leaving a sugary sauce off your, say, salmon or something like that. So the point is that, so we were looking at those, and one of the things we discovered is that just from the shape of the curve, everybody's curve's a little bit different. We can actually predict your subtype, meaning we could see whether you're muscle resistant or a beta cell defect just from the shape of that glucose curve. And then we went on to basically start characterizing what it means, to your point. And basically, we discovered that depending on your subtype determines the types of foods you spike to. So meaning we all spike to different foods. Some people spike to potatoes, some to pasta, some to bread, some to white bread, some to brown bread. Believe it or not, you can spike to very different things. And most people spike to white rice. In fact, little interesting fact, white rice is worse than ice cream for most people. So it's a very strong, if you will, glycemic food. It really spikes your glucose. Anyway, what we discovered is that as we subtype people and we looked at their food, how they spike the foods, we noticed that first of all, most people are rice spikers, as they say, but some are bread and this other. And it turns out, depending on your subtype, determines what food you'll spike to. So muscle resistant folks will spike to potatoes and pasta, whereas if you're sensitive, you won't. If you have a beta cell defect, you'll also spike to potatoes. So we're trying to match now. There's a lot more combinations to try here, but you get the idea that depending on the way your if you have glucose dysfunction, determines what foods you'll spike to. And we think this is also a big deal for the medication side too, because actually, I am a bit thin, so I I went to a trainer and I started lifting weights, and I gained 10 pounds of muscle mass. Took a while to do it, but it had zero effect on my glucose. And in the end, it made a lot of sense. It's because I have a beta cell defect. It doesn't matter how much muscle. The idea was muscle mass would be better for glucose homeostasis. But even though I gained lots of muscle mass, had zero effect on my glucose because I'm not muscle insulin resistant. It's not a muscle problem. It's a beta cell defect.
Yeah.
So I could gain, yeah. So you can really match these things. We've even taken it one step further, meaning it turns out for better or worse, I don't respond to metformin, which is the most common drug people at least used to use for type 2 diabetes. So, and I kept increasing dose, still didn't respond. I did try and control diet by diet, which worked initially at first, but then I finally, as it kept creeping up, did have to turn to drugs. And I'm a metformin non-responder, but it turns out once I figured out as a beta cell defect, I respond to it's a Prandtl derivative called rapinolide, and that actually I do respond to. So again, knowing the subtype of diabetes can determine what medication you should take and certainly what foods you should eat. So there's a lot more to work out here, but we are pushing that.
Yeah, that's fascinating. I think just to take a sort of a sidestep for a moment, I think that is a microcosm of my bigger issue with our current sick care system is that we are taking population data by and large and always applying it to the individual, even though we know the individual is so different genetically, from a metabolism standpoint, from a microbiome standpoint. And that's always been the case really up until really the advent of more personalized medicine, where we're just constantly taking large scale studies and then trying to prescribe therapies based on that to somebody who, in your case, doesn't respond and is never going to respond because it doesn't address the underlying pathophysiology.
Yeah, you're spot on. And that's the big theme of our work too. We're trying to get deep profiles, understand what people, their subtype really is. And if you think about all complex diseases, of which diabetes is one, I mean, there are what are called monogenic forms, single gene mutations that can cause disease. A good example is like BRCA mutations causing breast cancer, right? Those do exist. But most diseases that people have, diabetes, coronary artery disease, even Alzheimer's and dementia, these are complex diseases. They're not single gene mutations. And it's a combination of things, probably both genetics and environment, actually. And for your lifespan, you may know your genetics only accounts for about 16% of your lifespan for most people, whereas your environmental and lifestyle factors are playing a bigger role. And I think that leads us all being quite different. We've got to figure this out if we want to treat people, precisely and in ways that really help themselves. This idea of taking drugs, and hoping one will work. That it's changed a little bit, but it wasn't so long ago an article came out saying the best drugs work only in 1 in 4 people. There are a lot of these neuropsychiatric drugs, and they're very hit or miss. And that's again because the conditions are complex.
Yeah.
And we feel that's very, very true for diabetes as well.
Yeah.
And so that's why we've dug in there. And of course, I have a strong personal interest in my situation. So
Yeah. Yeah, well, that's great. So let's unpack this study. It was published in Nature Biomedical Engineering, I think, late last year. Maybe the print edition was in August of this year, but I believe it came out online.
That's right. Yeah,
I read it at the end of last year. So let's talk through just the study design, the methodology, so that folks can understand how you ultimately discover these different, and we're calling them 4 subphenotypes, but it sounds like there may be others.
There's probably a few more too.
Yeah.
The most common ones, by the way, are the beta cell defect and the muscle insulin resistance. Yeah.
Yep. Yeah. So how did you sort of design this study, and then again, let's go through the methodology so that folks can understand how you discover these and what tools you use to come up with the data set.
Yeah, I guess what brought us into the study was me being a beta cell defect. Once I figured that out, and then saying, well, I'll bet there's a lot of other people like me. And it turns out South Asians are often thin diabetics as well. People don't realize that. And my hypothesis was a lot of those would be beta cell defects. It turns out that's not 100% right. Some of them are muscle insulin resistant. But just knowing that, people, I don't look diabetic, I don't fit the normal paradigm of muscle insulin resistant is the most common. And so we just thought we should look under the hood, start teasing out the subphenotypes, if you will. And the way you do it gets a little bit involved, so I won't get overly technical on this, but the standard thing that many people can appreciate is something called an OGTT assay, oral glucose tolerance test. You drink a shot of glucose and your sugar will go up in your blood. And then with these CGMs, basically, which measure interstitial fluid, so just under your skin, they'll measure glucose that way as well. And they're pretty related, your interstitial and your blood glucose. It's like a 5-minute lag, but otherwise they're quite similar. And so you can do this at home. You can take a shot of glucose and see how bad you'll spike. And again, a lot of people have good glucose control, don't spike so much, but many people are moderate spikers. They're probably pre-diabetic. And by the way, 90% of pre-diabetics don't know it. Throw a few facts as we go along here. 20% of diabetics don't know it, believe it or not. We think this is terrible because we are in a diabetes endemic, if you think about it, where
Absolutely.
it's getting worse and worse. Basically, half the population, when you add it up, 11.6% of people are diabetic, 33% are pre-diabetic. So nearly actually now 38, meaning half the U.S. population is either pre-diabetic or diabetic. And most of those pre-diabetics will become diabetic within 5 years. So we've got a huge, huge problem here. And so we've got to get it under control. The existing systems don't work. Treating everybody, telling people, oh, just, don't eat cake doesn't work. But when you wear these glucose monitors, we have studies showing that just wearing a glucose monitor, people will improve what's called their time in range, and they'll even lose weight, meaning, you can see, if you see what's spiking your glucose, very visual, so you can modify your behavior. But back to the study. So what we did was we typed people with this OGTT assay, which tells you how badly you're spiking, and the shape gives you a shape of the curve. And then we can subtype them other ways for muscle insulin resistant, where you basically hold the glucose steady and you see how things are responding. And then the incretin defective, it actually, again, gets a little bit complicated. These GLPs, these incretins, they're made by your gut. And so when you eat food, they get released from your gut and then spread. And you can tell then the difference between the incretin defect if you shoot your glucose in your vein versus you drink it straight up. That delta is called the incretin defect. So that one's not so hard to figure out. Anyway, you can type it these various ways. And so that's what we did. And we discovered, first of all, even though we had a very continuum of people's glucose levels as measured by something called hemoglobin A1C, we could see this continuum of levels, but their curves were very different. There are these glucose curves. And also, as I say, their subtypes turned out to be very, very different, meaning you'd see one person who's muscle insulin resistant, a little bit of beta cell, but fine on the other categories. Other people who would be hepatic, some meaning liver insulin resistant. And many people are a combination of these things. And so in the end, and this is where AI comes in. So in the end, for step one was we discovered everyone's different in the way their glucose dysregulation is occurring. And second, and by the way, a lot of these people are pre-diabetic or normal. So they're already losing, glucose control. And we think that's a perfect time to catch people, right? We want to be able to get it under control early. It's very hard to reverse disease. So anyway, and then when we had these glucose curves showing interesting patterns, we could use AI and machine learning. And I don't know how, strong the audience is for this, but basically it's a way of letting computers figure out from the shape of the curve which features correlate with what kinds of defect in glucose regulation you have, and you can pull it all together in a very sophisticated fashion. That's what we did initially with something called machine learning. Now we're doing something called deep learning, which is even more sophisticated. But it works for two of these things. You can actually, we can predict quite well your muscle insulin resistant and beta cell defect just from the shape of this glucose curve. And now you can buy that from a drugstore over the counter for like $50 or some number like that. I think it's $70 for two, something in that range. Yeah.
Yeah, it's fascinating. We definitely use a lot of CGMs at Wild Health, really for the reasons that you alluded to. There's a lot of information to be gained, even without the benefit of machine or deep learning. Just looking at how dynamic those blood sugar responses are to different kinds of eating patterns. So in this study, you used a standard oral glucose tolerance test, and then basically just allowed the machine learning to analyze the curve dynamics to
We did, yeah.
different, subphenotypes.
Yeah, but we did take it the one step further. Then we did it with a CGM,
Right.
not just the thing you do in the doctor's office as OGTT. And the power of that is, it turns out it's a better measurement because it follows you continuously.
Right.
And so you actually get better predictive value from the home CGM than you do this fancy, very expensive test you do in a physician's office. So it will be the future, we believe, to do this at home. And so we're trying to validate the study one more time, although we're pretty confident the signals are so strong. And then the plan is to get it out to the world as soon as possible, quite frankly, because we think this information is very, very important for how to treat people and their lifestyle.
Yeah, absolutely. Of the subphenotypes that you all identified, which ones at least are most reliably predicted right now using the algorithm?
Yeah, the two I mentioned were muscle insulin resistant and beta cell defect. We have some signal from incretin defects as well as hepatic insulin resistance, and we're trying to figure out the contribution from the microbiome as well. But they're not strongly predictive just yet. So stay tuned there. I think we need that, a few more, they call them features to the model, a few more measurements, if you will. And then I think we can get there. So we're still working pretty actively on that.
Yeah, that's incredibly interesting to think about applying this. So eventually you said these algorithms will be something that would be accessible on a commercial level to patients who can then, wear CGMs and decipher some of this information. Would they all need to be doing a standard glucose load at home? Is there a food-based version of this?
Yeah, great question. So in my world, they would just drink the shot of glucose. Now, if you're pretty strongly diabetic, your doctor would probably advise against that because they don't like to see you get these giant peaks. So you'd probably only do it once. It wouldn't be the end of the world. But I like it because it's more precise measurement.
Mm-hmm.
But as we mentioned earlier, people spike to different foods depending on your subphenotype. So meaning that it turns out, I mentioned earlier, that people who are muscle insulin resistant will spike to potatoes. And it turns out most people spike at some level to grapes because grapes are just like pure sugar. So it turns out you can do a potato to grapes ratio and figure out whether you're muscle insulin resistant or not. And that will actually work. Now, the complication is if we tell people to eat 35 grapes, do they grab big ones or little ones and what variety? So there'll be more imprecision. I was trained as a chemist, so that's why I like people drinking their glucose. But it's probably the case the average, Joe and Jane, Smith will probably do the grapes to potatoes.
Yeah, that's certainly, I think that's a lot more feasible for most people. I did an AI summary to try to calculate the volumes of food, I think based on the paper that came out and some of the other available data related to this. I think it came down to something like 2.75 cups. So basically 2 3/4 cups of potatoes. And then you said 35, is it 35 or 55 grapes?
I'd have to look it up, but it's in that range.
Somewhere in there.
Yeah, it is. Yeah. And the potatoes we used were sort of the hash brown kinds of potatoes too. So getting your potato type. Believe it or not, that's a study we're now running. We're going to test the difference between French fries, mashed potatoes, and baked potatoes, and see which one spikes you more.
Yeah, that's interesting.
Yeah.
Yeah, so I think you would basically do this. You would do one food, I guess, fast overnight, do one of these food challenges in the morning, and then I guess on a separate day, fast
Correct.
and do the grapes, or I guess that's the second one. And then it's the ratio of potatoes to grapes in terms of how high your blood sugar spikes to those two things. Is that how the ratio gets calculated?
Yeah, that's exactly right. Yeah, you should be fasted so you can see, precisely how you spike for,
Okay.
after you consume the food. And then, as you point out, you do both foods, and you look at the ratio. And again, if you're muscle resistant, you'll be a potato spiker. That potato number will go up pretty high compared to your grapes. Yeah. But whereas if you're insulin sensitive, it won't. It may spike a little bit, but not as much. And of course, if you have perfect glucose control, it won't spike at all. Yeah, and I can tell you another fun area in this whole thing that we're working on now. We're working on knowing what your subtype is. Obviously, I mentioned drugs earlier. It determines what drugs you might take, but we're working on trying to mitigate those spikes. So you may know you probably shouldn't eat your carbs first. You should eat your salad or your chicken or what have you. So the idea is eat your protein or fat or fiber before you eat your carbs. And so we've been running people through that. And same thing, discovered, it's early days still, but we did protein, fat, and fiber were the first three we tested, where the people consumed 10 grams 10 minutes before doing the rice, which again, rice spikes most people. So, and it turns out if you're muscle resistant, you don't respond as well to protein and fiber mitigators, but you will if you're sensitive. So knowing your subtype determines which mitigators might work for you and which ones won't. And so we're still working on that too. There's a lot of combinations to test here, but we think it's all figure outable because there's a lot of people out there who are all happy to take these tests and quite frankly want to know what's causing their glucose spikes. And obviously if you can reduce those spikes, there's studies now to show that if you can reduce these postprandial spikes, they call them, you will actually, you reduce your cardiovascular events. So we think this is a big deal. And so the goal would be to get everybody doing this. And these glucose monitors, they are so visual. Like I say, they just, you can't, a double negative coming here, but you can't not use this and not improve your diet. There's triple negative. You get the bottom line is you use these things, you will improve your diet because it's just so obvious.
I cannot emphasize that point enough. I mean, certainly personally, I was the first time I ever wore one, I was blown away. You mentioned rice. Yeah, rice is one of the worst things for me to eat from a blood sugar instability standpoint. I mean, I'd spike in an exaggerated way, and then I have lots of spikes later. Just in things you don't sense clinically, right? There are no symptoms associated with these giant glucose excursions. They just happen over and over and over again, unbeknownst to you.
They can make you sleepy,
Sure.
and so that was one aspect. But yeah, you're spot on.
Yeah.
And no, it's we think it's important to know this. I was going to say one other thing. We also make food these days based on yield, the food industry versus nutrition. And so white rice today is 8% protein, whereas ancient white rice is estimated to be 20% protein. There's some wild varieties out there that are more protein. So if you think about it, right, we should be eating those kinds of rice and not these high yield white rice that's bred to just be full of. So we should be looking into that. And I think I have not yet tested brown versus white rice, but I suspect it'll be a bit better because it has other stuff in it. At least on me, I haven't tested, but there are varieties out there. And I think we should, as a society, be looking back into that. Because, historically we were raised with this more natural form that probably has more protein. So once again, we hit ourselves with an overabundance of food and an overabundance of bad food.
Right. Yeah. Yeah, that's a really interesting concept. I was not aware that rice has a much lower protein content, but that certainly makes sense why it would be much more of a metabolic disaster these days than it would have been historically for humans. Yeah, wow. And I imagine that's probably true for lots of other products that we've developed over time. Is it's just it's becoming less nutritious, and as you said, it's increasing yield at the expense of nutrition, and then ultimately at the expense of our metabolic well-being.
Yeah, absolutely. And of course, ultra processed foods, which is we mostly talk about it from the carb standpoint, cakes, cookies, muffins, these, which are very sugary, very sugar rich, high glycemic. They're terrible for you, but it's not. It turns out ultra processed meats are a bit of a problem too. The study just came out recently showing ultra processed meats, a much higher incidence of cardiovascular disease and cardiovascular events. So, yeah, we really have to get back to eating properly.
Yeah, yeah, completely agree with that. So I don't want to pin you down with a specific timeline, but you said some of the technology in relation to the machine learning and CGM, that those algorithms are going to be commercially available at some point. Do you have a sense for when those might be?
I'm hoping next year, and there's a good chance we'll put it up there for free. We're just trying to duplicate it now. I want to be sure our algorithms are robust. There's a validation study running right now. So my goal would be to get it out next year, 2026.
That's awesome. That's very exciting. Yeah, certainly I think with the number of people wearing CGMs now for wellness and just health optimization, I think this technology is going to be an absolute game changer, because I think a lot of folks are going to learn much earlier in the course of disease that they've got problems that need to be addressed.
Yeah.
Uh,
And I think it's pretty simple, just again, you can still eat things you like that don't spike you. And I think you just have to know what they are.
yeah,
So our goal is, like you say, and these tests, by the way we ran them in a clinic, they cost thousands of dollars.
sure.
But now with the simple, CGM or grapes and potatoes, it'll be dirt cheap to do this stuff. And yeah, my goal is to get it out there free or very low cost. So that's the plan.
That's awesome. So then I guess I know that you said there are validation studies that are happening, and then what else is coming down the pipeline from a diabetes care? Are you all looking to other domains in terms of further phenotyping or, further diagnostics?
We are. So we're trying to add on other characteristics, genetic information. We all have different copies of, they're called amylase genes that break down your starch into glucose. And so we're looking at the genetic component. We're looking at the microbiome I mentioned earlier and trying to add a few other things into the model, as well as the mechanisms of what does it mean to be muscle insulin resistant? What's causing that? And so we have some clues there. So we're trying to understand that better, but that's one aspect. Understand the basis of diabetes, getting more features into the model. Looking more at the foods, just like I mentioned the potatoes, which potatoes better or worse for you. And, baked potato, mashed potato, or, French fries. Offhand, French fries sounds like it would be worse. Probably is because of the fat, which is not good for your arteries, as well as the carbs, which is not so good for your glucose control. But we'll measure both of those things and we'll see what the numbers are. You get very surprised sometimes. So anyway, we'll bring like rice and ice cream. Like you say, most people just assume ice cream is worse, but
Yeah.
it's not the case probably for most people. Rice is worse. So anyway, so we're doing a lot more studies like that. And this mitigation I mentioned, which is a big deal. Now here I'm 100% conflicted, but we have formed a company, January I, that actually you can either use glucose monitors. They can actually even predict your glucose from your smartwatch and other features. And from that, they can make these predictions. They have behavior modification programs. Now, some of this stuff is generic and pretty obvious. Like if you should have your first big meal in the morning as opposed to late in the evening, and don't eat right before you go to bed, especially sugary things. So try to give yourselves 3 hours before you go to sleep, and that works. If you are going to eat something that spikes you, and this is what January will show you visually, do a brisk 15-minute walk and you will suppress that spike. So this idea of taking a walk after a big meal, this is not wives' tale. This is real. You should really do this. So if you are going to eat that bad thing, go take your walk after the meal. And so we're doing a lot more of these lifestyle interventions. I mentioned the mitigation stuff. We want to see dosing with the fibers and the fats and the other sorts of things. And there are many different kinds of fibers. This is where it gets complicated, but it's all, if you will, something we can decipher just by running the right tests and experiments. And so at the end of the day, it might be that it's known that vinegar can help suppress these spikes. So maybe that vinegar on your salad is what people who are at high risk should be doing before they eat any carbs in their meal. In fact, I'm sure it's true. That will help them. So these are the kinds of, but it'd be nice to know who it helps and who it doesn't help.
Yeah, absolutely.
You know what I mean?
Yeah, and I think it's probably worth emphasizing. I mean, we create these artificial cut points in medicine and science, largely probably because we need to be able to study things and speak the same language. But type 2 diabetes is a continuous spectrum. Prediabetes doesn't actually start at a certain A1C, right? The pathophysiology is there long before you get to that point. And the studies the data that you have collected already, you're collecting this in people who are not overtly diabetic, right? You have people who are seemingly healthy, who are somewhere along their pre-diabetic spectrum, and this technology works even in those folks.
Absolutely. Yeah. And we think it's important to discover it as before in these folks so they can do something about it while they're healthy.
Yeah.
100% agree.
Well, I think all of this stuff is fascinating. I imagine that most of our listeners are probably totally unaware of this. As I said, I've been bringing this up to colleagues of mine, certainly outside of Wild Health and, kind of in the traditional conventional medical space, and are totally unaware of this discovery that we have all these various subphenotypes of diabetes. And I think certainly explains why so many people fail traditional treatment strategies, because we're not addressing the underlying mechanisms. We could probably go on for hours about this stuff, but I certainly want to be respectful of your time. I know you have other commitments today. Do you can folks find you somewhere? Do you have anything that's public facing, social media, that sort of thing? Or
yeah, it's probably easiest to email me, M as in Michael, P as in Paul, Snyder at stanford.edu. Happy to take that way. I just have a Twitter account and a LinkedIn, but I don't really respond much to my LinkedIn too much. They get a little overwhelmed. So yeah, and but I'll just want to put a button on what you just said. We call this precision diabetes. We really want to nail it precisely. So, and I think that's going to be so important. So feel free to reach out, and happy to respond to your listeners.
Well, I can tell you from personal experience that you absolutely do graciously respond. I think that in some ways you're unique in that regard. I think for somebody of your academic stature, with all the various things that you do, with all the commitments, that you're still willing to take time to reach out to folks like me who are in the trenches providing clinical care with patients. And I think the impact that you and your lab have had and will continue to have on healthcare is impossible to quantify at this point in time. And I think we are, especially with the use of AI technologies now, able to harness big data in a way that we've never been able to do before. And I think it's going to revolutionize healthcare going forward. And I'm just super grateful for your time, of your willingness to share information. And my hope is that at some point we'll be able to have you back on the podcast and maybe have a personalized medicine series by Dr. Snyder.
Sure. Anyway, my pleasure being here. Thank you for the kind words.
Absolutely. I hope you have a great day.
Thanks so much for listening to the Wild Health Podcast. If you want more daily health education, be sure to follow us on Instagram, which is filled with health optimization how-tos, grocery shopping lists, guided breathwork posts, myth debunking, and so much more. You can find us at Wild Health MD on Instagram.